Hi, Dr. Rudin. Thank you so much for joining us today. Dr. Charlie Rudin is with us from Memorial Sloan Kettering Cancer Center in New York City, and we're here to talk about his research today. Dr. Rudin, you have really dedicated all of your career to small cell lung cancer. So tell us a little bit about that and what really drew your interest to small cell in the beginning.
Yeah, well, it's a pleasure to be here with you all, for sure. I guess I didn't intend to go into small cell lung cancer somehow. I was a thoracic oncologist, and early on was studying a lot of different stuff. Really, the opportunity to interrogate small cell particularly came up. There's a big sequencing project called the TCGA, which was a national effort to sequence lots of cancer types. And that list did not include small cell lung cancer. And of course, as we all know, small cell lung cancer is a major killer. So I was at Johns Hopkins at the time, and we saw an opportunity maybe to sequence the genome of small cell. So we had a number of samples that the surgeons at Hopkins had collected over decades, and thinking ahead, they had actually fresh frozen these samples as soon as they came out of the patients, and we had quite a collection of these, and so we partnered up actually with a drug company, with Genentech. They had the sequencing capacity, we had the tumors, so it was a natural synergy, and we sent them the DNA anonymized, and they did the sequencing, and so we published the first small cell genome study in about 2012, and there were very few people working on small cell at the time, so suddenly we were like the experts, which was ridiculous, but it was true, and so we had characterized the genome and the alterations in small cell. That really got us started. And then the next year, 2013, Harold Varmus, who was the head of the NCI at the time, reached out to me and to John Minna. He had seen our paper on the genome. And he said, we want to have you guys lead a national effort to write a white paper for Congress to get funding for small cell as a recalcitrant cancer. And John and I kind of invited everyone we could think of around the country to come advise on this, because there weren't very many people working on small cell. But we did. We put together this white paper, it went to Congress, and it became the foundation for what became the NCI Small Cell Lung Cancer Consortium, which is a group of laboratories around the world, not so much clinical, but preclinical, studying small cell. And over the last now more than a decade, this has really driven a lot of preclinical research in small cell that has led over that time to new insights into the disease and to new therapies for the disease. So that got us started. And it really wasn't a plan. It was just, no one had sequenced this tumor, and people were dying of it, and it seemed like we needed to take it on.
Thanks, Dr. Rudin, for highlighting that, and welcome to our smashers who are listening in.
Hey, thanks.
This is just going to be such an amazing chat today. I know, Anne and I have both fangirled on this one, and we're looking forward to all the discussions. I know, really kind of focusing on trials, but as a thoracic medical oncologist, as the really the thoracic leader for Memorial Sloan Kettering, one of the top cancer centers in the world, and as one of the absolute top small cell researchers, we're excited to pick your brain. Can you give us a little history lesson of what did small cell treatment look like 10, 20, 30 years ago, before all the exciting changes with the Recalcitrant Act and all of your research? What did it look like maybe in the 80s or 90s for patients who are impacted by small cell?
Yeah, 80s, 90s, 20, turn of the century, even the tens, it was all pretty much the same as, Dr. Shields. It was really platinum etoposide and then maybe topotecan. And then we really didn't have anything. It was pretty simple. You could train a small cell clinical expert in about 5 minutes because there weren't a lot of drugs. It was a pretty simple paradigm, and for limited stage, it was the same drugs, platinum etoposide with radiation, and that was it. So we didn't have much, and there were all kinds of chemo that people had tried, of course, and there were other regimens, but there was nothing better than that. And it was quite. It was really a death sentence at the time to get this disease, and that's no longer true, of course. But that was pretty much the state of the state.
Dr. Rudin, kind of to follow up on that, why do you think that progress for small cells seemed to stall over that time? Why was it such a difficult cancer to treat?
Yeah, I guess there was a sense of nihilism about it, and as we all know, this is a disease that's strongly associated with tobacco use in most folks, and I think there was a stigma around that. And I also think there was a sense that in the 80s, maybe in the 90s, before I was really active in the field, but there was a sense that this was going to be an easy one. We were going to cure this. It was sort of the leukemia of solid tumor world, and seemed like aggressive chemotherapy should kill this, and it didn't work, and many careers sort of died on the shoals of that graveyard, and trying with different chemo regimens, and then I think people kind of gave up, and there was a sense that there was super excitement in the non-small cell lung cancer world in lung cancer, because there was the discovery of the driver mutations and EGFR and 2004, this discovery of that there are these mutations that are targetable, and one after another, it was clear that lung adenocarcinoma was going to have these driver pathways that were targetable. Most of them were kinases, and we could drug them. And I think everyone in thoracic oncology sort of pivoted and said, "Wow, this is where the action is," and that's where the action was. And we made a lot of progress there. But I at that time was sort of thinking, I wanted to do something that other people weren't doing, because it was a crowded field, in the adenocarcinoma world. No one was really working on small cell. So it was a real opportunity, at a time when there weren't, competing folks really trying this. And so it seemed like an area where we could make more impact than doing
Thank you.
what everybody else is doing.
And thank you for your courage and your pioneer, nature, and to really trek through a field that had had so many, failed opportunities and failed options, and you have led the way to provide hope to so many, and lives changed, and time gained. One additional kind of question here is, as a physician scientist, you run one of the most prominent small cell lung cancer labs in the world. What during that time brought you maybe frustration? I think you touched a little bit on the nihilism. Was it funding? Was it resources? Was it insight? Was it people caring, knowledge? What were some frustrations that you were experiencing for that time, for any of our researchers that might be listening in?
Yeah, well, first of all, it's a team sport. I mean, I was not leading. It was a group effort for sure, and I learned way more from the people working in the lab than I teach them. I mean, they're a bright young group, and that's the fun of the job, is sort of the learning from people who are active at the bench and active in the clinic. Every bit of it is a team sport for sure. Frustrations, funding was a problem, and that's true across the board for lung cancer, but particularly for small cell. And I do think this NCI initiative changed that pretty dramatically. There were almost no R01s for small cell before this consortium started. And when Harold Varmus sort of prompted John and I to take this on, he said, this may not lead to any funding. I just want to be clear with you guys. This may not lead to anything. But we should do this, and it did, So that was definitely, a frustration. But at the same time, working in a group in a field where there weren't a lot of other players meant that there was a real team sense. Nobody was trying to hide anything. Everyone was trying to get their stuff out there and share. And so I think, the small cell community has grown up, as Dr. Shields. It's a very collegial group and a pretty actively engaged group of sharing data and sharing credit, and trying to make sure credit's offered where it's due. And I think it still has that spirit, even though now we're quite a larger community.
I fully agree. I think it's just, I know Anne can attest to it as being an oncology nurse for goodness, 30 years?
Longer than that.
And myself, as you mentioned, Dr. Minna, yourself, and so many of the others who have really led this field are just so grateful and gracious and kind to give back and to support and collaborate. And I 1000% echo. This is just a lovely group of researchers. And I think our smashers have seen that.
And of course, you were growing up in his lab at that time.
Yeah. So, you're right. I have been doing this for a long time. So I really saw that very slow progress in the small cell community, and you're right. We saw so much hope, and sort of we got excited in the non-small cell world there, in the late 90s and into the early 2000s. And, I kind of felt at that time we were a little bit in our heyday, but still nothing was happening in small cell. So what actually gave you hope during that time to kind of keep pushing and keep going in small cell?
Well, I do think, there's a sense that this is a tide that raises all boats. We started to see progress in lung adenocarcinoma, which was also a lethal disease. And as oncology took on a new phase of really thinking about targeted therapies, identifying the drivers of the disease, identifying the vulnerabilities, we had new tools in the toolbox to begin to interrogate this, and it just seemed like a good time to refresh the efforts in small cell, not just throwing another poison at it, which was sort of the approach with chemo, was find a drug that's really toxic and see if we can get a therapeutic window. Instead of doing that, really trying to figure out the biology of the disease and go after that, I think gave hope across the area of oncology, not just small cell, but, you see the progress in diseases like, I don't know, multiple myeloma, another, totally lethal disease, now basically manageable. Of course, there's still lots of folks that unfortunately succumb to these diseases, but I think the tide was turning, and the tide was, the time was right for it to address small cell as well.
And thank you for that insight. And to just kind of carry on with that whole theme, when in your own research, at what point did you really kind of start to have an aha moment and think that this tide was turning? And, was it in sequencing? Was it in modeling? Was it in the biology of the, what actually did you had that gave you that aha moment? Like, this is, I'm learning something that's good. Let's keep going.
Yeah, I think probably the real thing that changed the tenor of our understanding of the disease and its potential for curability was actually the advent of immunotherapy. You know, even with the targeted therapies we were looking at, those survival curves tended to go to ground. You know, there weren't long-term survivors across the board, but immunotherapy raised the tail of the survival curve, and seeing that in small cell, that there are patients with metastatic disease whose disease was controlled long term. You know, I have a patient who was treated with small cell in one of the first clinical trials of immunotherapy for small cell. She's out more than 15, close to 20 years now. She had
Thanks.
metastatic disease to the brain, and those patients are rare, but they're a benchmark. They're such an important thing that with immunotherapy, with training the immune system to recognize this cancer, that long-term survival is possible. Just understanding that that's possible, I think was a game changer for me and a real eye opener. Because you know, when I was training, if we had a patient with small cell lung cancer, the goal was quality of life. The goal was not cure. And if we have long-term survivors, then it changes the nature of the goal. It changes what our goal is. Our goal should be cure.
That's a that is just a great point, which really leads into talking about clinical trials. And, going back even 10 years ago, 15 years ago, the clinical trials that we had for small cell were so limited. And I think that especially during that time, as you spoke about nihilism, I think our patients really felt like in small cell, especially whether they were extensive stage or limited disease, if you were offering them a clinical trial, that must be the last resort. There must be absolutely nothing else you can do. So, can you speak a little bit about how that sort of attitude around clinical trials has changed, and what we're seeing in clinical trials in small cell, and even going into first line therapy trials now in small cell, which is just phenomenal. It's really fantastic.
Yeah. So I do think, clinical trials offer patients the opportunity to benefit from what I think is sort of the cutting edge of understanding of the diseases, including small cell lung cancer. So uh, it is true that historically, drug development, when we were just sort of uh, throwing poisons at this disease, as I was saying, that it was sort of a last gasp effort in patients who didn't have other options. Now I think the tables have really turned on that. I would say that um, the opportunity to participate in clinical trials offers patients the potential to benefit from our current understanding of the disease. Many of those trials, unfortunately, won't benefit the patients, but we learn a tremendous amount from those. Even the trials that are unsuccessful, we learn from. And I have led a lot of trials that unfortunately didn't work. International phase 3 trial looking at this immunotherapy TIGIT that people were really excited about didn't work. But understanding that is going to benefit the field, benefit future patients. So I think it offers two opportunities. One is direct benefit to the participant from potentially getting what might be the therapy of tomorrow today. And then the second benefit is to the global community.
Yeah, I think that one of the strongest points that I always try to make about clinical trials is that every standard of care that we have today came from a clinical trial. And without our brave patients who consented to these new and cutting edge therapies that we offered, we wouldn't have the standards of care that we have today. So, we look at the things that you've mentioned it several times. I mean, look what the clinical trials in immunotherapy did for small cell. and now we're at Lurby and Tarla, and now we're getting into the bispecifics. So can you just share a little bit about what your thoughts are around those trials and what trials we're looking at right now, and what our small cell smashers might have to look forward to that's coming down the pipe?
Yeah, there's a ton of new drugs now in small cells. It's a super exciting era now for trialists and for patients, because there's real drugs that are showing real efficacy in this disease. And that's great to see. And I think more and more drug companies are developing drugs in this space. And there are multiple that look quite interesting. I would say, first of all, academic oncologists like Dr. Shields and myself, we try to, and the drug companies too, try to make sure that if we're introducing a new drug in this space, we're trying to make sure we're not doing patients harm. And so we want to make sure patients have at least the opportunity to get the standard of care, maybe along with a novel agent, or if they're getting a novel agent, that they have the opportunity to then, cross over to get a standard of care. And every trial, is designed with a pretty detailed ethical review to make sure that it's, valid and reflective of good clinical practice. But some of the drugs that are really exciting to me, I think, the antibody drug conjugates are showing real activity here. These are basically antibodies that are directed against the cancer that bring in a payload that's basically a chemotherapy, but it directs it to the tumor. So it's a little bit of a smart bomb. You're directing the chemo directly to the tumor, trying to avoid delivering it to all the rest of the cells of the body. So it's a way of focusing the cytotoxic effect of chemotherapy on the cancer and avoiding some of the toxicities of standard chemotherapy. Those are showing real efficacy. So there are multiple dozens now of antibody drug conjugates that I think are looking, maybe not dozens, but certainly a handful that are looking really promising in small cell lung cancer right now. So that's a really exciting area. The other class of drugs that's been really exciting are these T-cell engagers. So these are drugs, again, sort of like an antibody that binds to the tumor, but it also binds to the T cell, which is the immune cell that is responsible for killing bad cells in the body, essentially, eliminating bad cells in the body. Often, I think, for infected cells, but also for cancer cells. These bispecifics basically bring in the immune system into the tumor and bring the cancer cell together with the immune cell, and the immune cell can then kill cancer. These have been really effective. The drug tarlatamab that's now FDA approved is in that class. And there are multiple of those also in clinical development right now. And tarlatamab is certainly the first out of the gate for those, but it may not be the best of the class, and there's other ones that look pretty interesting and that I think have real promise. So those are the two classes of drugs I would say are super promising right now, are T-cell engagers, antibody drug conjugates. And then there's a whole slew of other stuff that's really exciting, but that is maybe earlier in its development. Radiotherapeutics, where they're conjugating a radiation to an antibody or to a small molecule that, again, delivers it to the tumor in a very directed way. CAR T cells that are T cells kind of programmed to recognize the cancer, and a whole slew of other stuff that's cooking. So I do think this is a time when there's so much going on. Not every drug is going to be a success, but there's clear evidence of highly effective drugs in development. We need to manage the toxicities. We need to make sure that they're safe, and that we're giving the right dose, and that we're managing the adverse effects properly. But these are going to be um, uh, I think, game changers. They're going to be game changers among the drugs currently in development. And I think the standards of care are going to change, within the foreseeable future, meaning 3 to 5 years, not 10 years.
That's really exciting. And I take that back to the patient that you just talked to us about that went on one of the first clinical trials for immunotherapy. And I mean, even she can look back now and say that she was part of a clinical trial that literally changed the standard of care. So
Yeah.
what you're saying about changing our standards of care in this disease, in the next, 5 to 10 years, I think it's just really exciting to think how different that's going to look.
Yeah. I
Absolutely. And
totally agree.
thank you for such a wealth of information and really a beautiful overview of all the exciting drugs that are being studied. And, one thing that I think as oncologists is knowing that sometimes these certain therapies may not work for all patients, right? You
Yeah.
mentioned, one therapy that didn't seem to benefit with TIGIT. Um, can you speak to kind of maybe some other things that might stand in the way of a therapy working, like plasticity or heterogeneity or neuroendocrine, non-neuroendocrine? Can you kind of speak to that for our smashers and what that might mean and why certain therapies may work for certain patients and not for others?
Yeah. I do think, small cell lung cancer's got a characteristic that we call plasticity. And a lot of us have been interested in this. This is sort of a shape shifting phenomenon where cancers have one appearance, but then if you're treating that cancer, it can shift to look a little bit different and avoid the toxicity of the therapy by that. So the cancer is kind of smart. The cancer is kind of slippery, and it shifts shape and shifts its appearance. And so, one of the things that investigators around the country and around the world have sort of been looking at is both in mouse models of small cell lung cancer and in patients with small cell lung cancer, trying to understand what are the dominant states that the small cell lung cancer exists in, and what do those really look like, and what are the drivers for those states. And I do think we have at least a first handle on the predominant states that these cancers can occupy. And understanding that's really important because those states, if the cancer shifts from one state to another state, understanding what the targets are in those different states can help us guide therapy. Because there are patients whose tumors predominantly look like one state and tumors that look like a different state. And those patients may need different therapies. So treating all patients the same has sort of been our approach in small cell, but I think there is at least an opportunity to begin looking at, are there ways of doing a little bit like we've done in non-small cell lung cancer? EGFR patients get treated with an EGFR inhibitor, and KRAS patients get treated with a KRAS inhibitor. I don't think it's going to be that easy for small cell, honestly, because of this plasticity, the ability of the tumor to shift from one to the other. But I think understanding the drivers of each of these states will give us a handle on approaches to drug development that would be specific to these states. And that may be a real way forward. I would say that's not in the 3-year period. That's more of a later horizon, at least currently. But it's an area of real interest. Your lab, my lab, other labs that are really interested in this.
Thank you, Dr. Rudin. And just for our smashers listening in, just to give a little bit more context, Dr. Rudin really led the pivotal studies that introduce neuroendocrine subtypes into small cell. He did the first study of getting this in patients and really classifying neuroendocrine subtypes, really pioneering the way yet again for patients and their families
Yet
impacted
again,
by
team
small cell.
effort.
Yet again, yes.
Team
Um,
effort. Team effort.
So I'd love
Lots
to
of
pick your
folks.
brain, Dr. Rudin, as
Yeah.
the world expert on subtypes. What do you think these biomarkers, such as the subtypes, play a role? And do you think that we'll use subtypes in testing for small cell?
I mean, I certainly hope so. I think it's one avenue of pursuit that I think it could lead to clinical benefit. I would say for our patients today, we don't really have a handle on how to use those appropriately and how to give best therapy based on the subtypes. We're still collecting a lot of data in that realm and trying to understand how can we constrain the tumor from changing state? Is there a way to constrain plasticity? And will that lead to a better approach to treating? I would say this is in the realm of preclinical research, um, where we don't quite know how to translate it clinically. There are ongoing efforts to try to do some initial clinical trials based on subtype. There's a national trial that Ann Chang at Yale is leading, that I think is really interesting to try to use profiling prospectively to enroll patients onto a therapy that's sort of dedicated toward their predominant subtype. Um, we'll see if that works. And I think, that's going to be an interesting trial. I am concerned that the tumor is going to be slippery and escape. And so, um, in my group, we're still kind of trying to figure out, are there ways that we can prevent the slipperiness, if you will, prevent the plasticity, and sort of lock tumors into one state where I think we can wrap our arms around it a little bit more. So I'm hopeful that that may lead to therapy advances, but I think we should all be a little bit agnostic about what's going to work, and we need to see it work.
We've talked a lot about the clinical trials and these changing paradigms in the treatment of small cell lung cancer. And for our smashers that are listening to us today and contemplating whether they might be eligible for a clinical trial or whether they should actually approach their treatment team about a clinical trial, what questions would you tell our smashers that are listening that they should ask their oncologist? What should they know about clinical trials, even the subtypes? I know that this subject has come up over and over, and our smashers are actually a group of very well read, intelligent patients who are doing their own research
Yeah.
on their illnesses. So when you talk about these trials for the subtypes and going into first line studies, what should our smashers be asking, and what do they need to know about enrolling in these clinical trials?
Yeah. I would say the clinical trials that I think are most exciting right now, those drugs that I was talking about, the antibody drug conjugates, the T cell engagers, and others, those don't tend to be subtype specific. So I think my advice to patients at this time would be to not be as concerned about the subtype, honestly, even though, it's an area that I'm really, really interested in and that our lab is really working on hard. I think it's just not quite right for clinical translation now. And I think the most exciting drugs are these other ones that are actually crossing subtypes. And that's a good thing. We want to be able to treat across subtypes. And so I think that would be a first message. I think, I would talk to your doctor about the opportunities that exist. Are there trials that would be appropriate, either within the oncology clinic that you're at or locally available to you? I think there's a balance that needs to be struck between trying to get to the most innovative clinical trial and, practicalities of life. We all want to live at home. We all want to live with our family. I think those support structures are really important. I don't encourage people to travel around the world to do some trial if it's going to completely remove them from their life and their support system and their and all of the things that sort of contribute to us having a good life. So I do think you need to take into consideration multiple factors, and all of our lives are complicated, and there's people that we love and we don't want to abandon, and you need to balance quality of life with uh, trying to get good care. So, it's a complicated answer, honestly. I think my advice to patients would be seek out opportunities, get as long a menu as you can, because the more things you have on the menu, the better. And then you can sort of sort through what makes sense. What makes sense for you? What makes sense for your family? Clinical trial participation, I think, does offer opportunities, as we said. It does also, patients on clinical trials are really closely monitored. There's a lot of visits. Often there's additional blood draws and other things. And um, so, you need to sort of take into consideration, can I get to New York on a weekly basis for a year or not? And things like that. So, uh, I would encourage people to seek out the information and the opportunities, and then, sit down and think about what makes sense.
That is really great advice. And I know that I'm sure you hear this from your patients. I know Dr. Shields hears it from her patients. We certainly get these questions a lot at LUNGevity is, what can I do that will accelerate this research? What can I
Mhm.
do as a small cell patient that will make this go faster? So do you have any advice there or things that you would say to a patient about the importance of participating in research?
Well, I do think we learn from every trial. And so, if there's an opportunity to participate in a clinical trial and it makes sense for you, I would absolutely encourage that. I do think that's how we make progress in the field, and our patients are 100% our partners in doing that. We can't make progress without the patient involvement and active participation. So I think the best way that patients can contribute is by thinking about the trials that make sense for them, and if it does make sense, to consider participating in that.
And Dr. Rudin, kind of to follow up on that, just to clarify for our smashers who are listening in, not all trials are treatment trials, right? Can you speak to, what we call non-interventional studies? So our listeners have a better understanding of that if they're seen on clinicaltrials.gov and seeing what that might entail.
Yeah, I mean, I think there are multiple trials that are not necessarily looking at therapy, but that are studying aspects of the disease. We recently did a trial that was looking at PET scanning, looking for DLL3 PET. So DLL3 is a target for a number of drugs, including tarlatamab and now some antibody drug conjugates and other things. But to understand if your tumor has DLL3 as a target would require biopsies, and that's not easy, and certainly not without risk. And so we developed a PET scan that could actually show DLL3 on the surface of tumors, and we've enrolled about 70 patients on that. Not only small cell, but neuroendocrine prostate cancer, kids with neuroblastoma, all kinds of different folks, to look for DLL3 expression on the tumor. Um, there's no therapy associated with that, but I think it's going to be a really interesting way, a non-invasive way of understanding the tumor target and whether a therapy is appropriate for those patients. So trials like that can be really helpful for the field, even if they're not, looking at a new drug.
I think that this really is so helpful, and I know that we have several patients that also are enrolled in longitudinal programs where they're being followed, or
Hmm.
even, potentially donating their body to research if they were to pass from small cell. Can you speak to how those studies might be helpful to advance science for understanding small cell?
Sure. I mean, that sort of brings to mind a case that we had, a wonderful 19-year-old kid who had small cell, and small cell doesn't really happen in 19-year-olds very often. And he was a never smoker, and he was a medical student. Was going to be a medical student. He was pre-med at Duke when he developed this rare small cell, and we were intrigued and confused by him, because why did this kid get small cell lung cancer? And that led us down a path to really discover this new disease entity, which we're calling atypical small cell, which doesn't occur it's not tobacco related, and it ends up with very much the same disease, but through a very different pathway. He graduated from college. He made it through. He lived a number of years with his disease, getting treatment both at Duke and with us at Memorial. And at the end of the day, he volunteered as a volunteer at Sloan Kettering, advising other patients and helping out around the cancer center. Just a lovely, lovely guy. And as his disease was unfortunately escaping from us, he decided that he was interested in medicine, he was interested in research, and he dedicated his body to
Mhm.
to the cause. And I give him a ton of credit for that, and not an easy decision, but he did, and that really led us down this path to figuring out this type of small cell that occurs in never smokers. We wouldn't have found it without him, and it was really because of his generosity uh, to the field that we've discovered this, and we've continued to work on that, and this is rare. It's maybe 3% of small cell, but it's a really interesting and important group of never smokers that get small cell, and can get it at any age. We've seen it in a 15-year-old as well. So um, I think those sorts of efforts can lead us to a deeper understanding of disease that we otherwise couldn't get. His
Yeah,
name's Khalid, and he's just a wonderful kid. I miss
when
him.
I read his story, I think it may have been in one of Memorial's newsletters or something, but I read his story, and it really brought home to me, because Mark Szynski and I had one of those over 20 years ago, and she was a nursing
Mm.
school student, and her actual they were doing respiratory assessment, and one of her you know classmates actually listened to her lungs and heard what she thought was RALS, and asked the instructor to come over and listen, and they sent her for a chest x-ray. And at that time, 20 years ago, Mark Szynski actually sent her path out to a specialist in Scottsdale, and he called Mark and said, why did you send this to me? This is straightforward small cell lung cancer. And we said, wait till you hear this story. So as soon as I
Yeah.
read that story about your patient, I thought, wow, so little did we know back then. So little did we know. And it makes me wonder, like, is she one and the same? She was
He must have been. I would bet on
she
it.
was 19 years
And
old as well, and in nursing school. And yeah.
yeah. And everybody has a case or two like that, and they kind of throw up their hands and say, "We don't understand." But when we found him, we went back and retrospectively, we found at least 30 of these in at Memorial. So they're out there,
Wow.
And, there's probably people online with us right now that have this.
That are young and have never
So
smoked.
Do you
it's
know,
rare, but it's important.
Dr. Rudin, to follow up on that, is there a trial that's looking at options for this particular atypical small cell?
Well, interestingly, they're to get into the weeds a little bit. They're locked into this ASCL1 state, so they have. We talked about the plasticity and the ability of small cell to move around. They don't move around. They sort of have this one subtype. We don't understand why, but that subtype has very high levels of expression of the target DLL3. So I think, we've started to treat, if we see one of these, we treat them with tarlatamab or another DLL3 directed therapy. And so far, I think, that looks like a really good drug for those folks. So we don't have a trial specifically for them, but anecdotally, I think we know what to do with them. We're learning one by one by one.
So you're using tarlatamab in first line with these patients?
Oh, not first line, but
first line.
we try to get them to tarlatamab. Yeah.
And I know that we're coming up on time, and I just want to give a shout out to our smashers. If you have any questions, drop them in the chat. We'd love to hear from you.
Yeah, absolutely.
And pick Dr. Rudin's brain some more. I do have one clarifying question, about patients who have been impacted by small cell and may have brain metastases. How do they pursue trials? Are there unique criteria or circumstances they should or shouldn't do for trials?
Yeah, I think increasingly we're pushing for trials to be open to patients with asymptomatic brain metastases, because what's become clear in the study of some of these new drugs is the response in the brain is not that different than the response in the rest of the body for many of the new drugs. And so enrolling patients who have asymptomatic brain metastases makes good sense. I think for a patient that has brain metastases, it's important to get a multidisciplinary view of what's the best approach to that, because some brain metastases really should get some stereotactic radiation to treat them. Some may need surgical resection if they're, in a position where really important structures in the brain are going to be affected. And some are probably safe to go on to drug-based therapy, systemic therapy, and even participate in clinical trials. But I think the details for that are kind of complicated. And so getting a multidisciplinary view by a medical oncologist, a radiation oncologist, and a surgeon for how to best manage them is important. But I do think brain metastases does not mean you can't participate in the clinical trial. Brain metastases means we need to deal with that, and we might have to deal with it a little bit separately than we deal with the rest of your disease. But we need to deal with it and get it taken care of, and then be able to look at the options.
Thank you so much, Dr. Rudin, for joining us today. We'll close out by asking you the question that we ask all our guests on our Fireside Chats, and that as you look back over your career, what gives you the most hope for small cell lung cancer patients and their families today?
Yeah, I think the diversity of new approaches that are now being looked at for this disease really gives me hope. There's real momentum that comes with that, and not having more shots on goal means we're going to get more therapies that work. And for a long time, honestly, drug companies weren't even taking shots on goal. And now they really are. And I think there's a feed forward loop that comes with understanding of the disease, defining targets, and drug companies becoming invested and interested, and then that leading to better understanding of the disease and new targets, and this sort of snowballs. So I think we're in that phase of accelerating progress in this disease, and I'm just thrilled that I'm still around to watch it at this point in my career, because it's really exciting. And I think the next generation of oncologists, Dr. Shields, I would put in the next generation, are really gonna see this, really change people's fate.
Well, I know that Ann and I are so grateful for this conversation today. I'm sure that our smashers listening in, any researchers, any physicians listening in have learned so much from this conversation. And the progress that we're seeing, the hope that we're seeing in small cell today is because we get to stand on the shoulders of giants like yourself, Dr. Rudin, and all the advancements and advocacy and progress and research and boots on the ground when no one else was there for our patients and their loved ones to make this progress happen. And you're seeing the fruits of your labor today. And I know that our patients and their loved ones are thankful for all that you do. So I think with that, Ann, we'll wrap up. I'll turn it back to you for any announcements for next time. And goodness, thank you, Dr. Rudin, for all you do.
Absolutely. It's a pleasure. And thank you for those kind words.
Thank you. I add my thanks to Dr. Shields. We appreciate all that you do for all of our lung cancer patients, and also just how much you support us here at LUNGevity Foundation. We appreciate all that you do. We are going to take the month of July off, so we will see you again on August the 28th, when our topic will be our hope, and we're going to really dig into some of these newer therapies that Dr. Rudin has introduced us today, and that will be our discussion in August. So thank you so much for joining us, everyone. It's been a pleasure to have this conversation with Dr. Rudin, and as always, I love all my conversations with my co-host, Dr. Misty Shields. So everyone have a wonderful weekend, and we will see you again in August.
Thank you. Thank you. Happy Friday.
Take care. Happy Friday.